other related topics in this anti-cancer realm that we're focusing on. So a reminder about upcoming deadlines. So you do have your annotated bibliographies, which I've given you guys an extension just because I want to make sure that they are at their best, and also that you have thoroughly read the material so that you get a sense of whether you feel like you're on the right track in terms of the material that you're looking at, or perhaps through the actual bibliography, I can give you some suggestions to the different directions that you can go. In addition, those that are presenting next week, I do need your articles so that I can distribute them to the rest of the class. All right, so what we will do today, we're going to continue our discussion about Platinum 2 with regards to its anti-cancer activity, but also pointing out some of the issues with regards to the use of this metal, specifically having to do with its off-target effects. Then we are going to transition to discussions of other metals that are being applied in an anti-cancer fashion. All right, so let me go ahead and project. All right, so first I will start off with a bit of a review of things that we have already discussed last time, just to reorient ourselves. So in our discussion, we talked about cisplan undergoing extensive changes in speciation even before it reaches the cellular environment. So here, this depiction of cisplan as if it remains intact maybe is a bit misleading. It's not entirely clear. Some portion of this compound can associate with the albumin protein. Some portion of this compound may actually remain a small molecule, but with changes of ligation having to do with dissociation of the chloride for possible aqua ligands binding or even carbonate. Nonetheless, it's understood that the delivery of this particular compound may go through two different fashions. One via a passive type diffusion, and the other via a transporter-assisted mechanism. So here we have the CRT1, which is a copper-based protein that can facilitate the uptake of this compound, and also the idea that the compound intact can be delivered into the cellular environment, and the changes in the concentration of chloride can lead to changes in the speciation of this compound. I also presented to you some issues concerning the acquired cellular resistance due to overexpression of proteins that can participate in the recognition of the platinum center, really the platinum compound, such as then to facilitate the release or the efflux of this compound. So here we have representation of two participating multi-drug resistant proteins, ATP7A and ATP7B, both copper trafficking transmembrane proteins that are believed to participate in the efflux of this compound. So there is an overexpression of these exporters. I also discussed that within the cellular environment, the platinum center can interact with the tripeptide glutathione, an important small molecule peptide that helps to regulate redox activity within the cellular environment. It is present at very high concentrations. We're talking about millimolar concentrations. And so via the thiol group, this particular peptide has the capacity to bind platinum being a soft metal. And so it can form adducts of this nature. And that can also be recognized by another multi-drug resistance protein, the MRP2, which can then facilitate the efflux of this compound. So that interaction of glutathione is seen to be something that can attenuate the activity of the platinum center. One of the major targets, and probably a bit misleading in terms of being the sole target of cisplan, is DNA. And so here we're focusing on where within the DNA molecule, platinum can actually have an effect. And so here I'm just highlighting the guanine nucleic acid that is believed to interact with the actual platinum. And so we're focusing on the nitrogen 7 of the base of this molecule that has the capacity to interact with the platinum. Now, for whatever reason, similar sites in the other bases don't have as much capacity to bind with the platinum. So that is not entirely clear to me. But nonetheless, the platinum is able to interact at this particular site of the base. And so there's speculation as to different types of metallation that can occur. You have proposed 1,2 intrastrand crosslinks, 1,3 intrastrand crosslinks. You even have suggestions of also interstrand crosslinks. And now that it's better understood the speciation of the platinum center, there's even suggestions that perhaps more monofunctional adducts, as opposed to these type of crosslinks, may be the mechanism by which the platinum can interact with the guanine of the of DNA. And so the idea is that however this platinum center interacts with DNA, what it does, it actually will alter the structure of this nucleic acid. And so its interaction, it seems to be as debilitating, as then affecting the capacity of the DNA to engage in replication. So there are other proteins that are responsible for the unwinding of the strands and then facilitating the ribonucleotides to then assist in the replication of the DNA molecule. One of the really principal one is topoisomerase, and so it's believed that this distortion of the DNA can affect that type of interaction. It's also believed that this distortion can lead to fragmentation of the DNA. And so here is an important study. This is an agarose gel that was performed in order to see what can happen to supercoiled DNA. So as I mentioned to you last time, here we're looking at the different types of effects that can occur. One type of effect is that the distortion in the structure of DNA can lead to a linearization, and that linearization can be perceived in terms of how the DNA runs in this particular type of gel, where if we look at the controls, a supercoiled versus a linearized DNA, so supercoiled is represented here by form 1, the linearized form 2, they may be of the same size, but the shape will dictate how they run through this gel. So form 1 runs further along than form 2. And so the presence of the platinum, and so the extent of platination, can lead to this linearization, so a distortion of the structure, and then also can lead to the formation of fragments, so basically can cleave the DNA. So here's another mechanism by which the platinum can affect the structure of DNA by literally breaking into pieces. But something that's very important and that contributes to the acquired resistance towards this drug is how the DNA repair mechanism can respond to the presence of platinum. So this is an additional form of resistance that has been observed, where you have an enhanced DNA machinery taking place to help remove the platination of DNA. And so there are already DNA repair mechanisms present in general, but these become enhanced. And so you have the role of the nucleotide excision repair, which can essentially help to cleave the interaction of the platinum with the DNA, thus enabling the DNA to adopt its, let's say, its endogenous or native form to facilitate the downstream replication. And so we combine the effect of the NRE plus the presence of those multi-drug resistance exporters, you have this sort of compound acquired resistance that shows why over time a drug like platinum, like cisplatin, can really lose its activity. And as we'll see in a moment, how there could be then off-target effects of this particular compound, or really the metal center. And so here, what I want to emphasize is that then it actually is more complex in terms of where exactly is platinum hitting, and are some of these non-DNA targets really off-target effects that can lead to toxicity. So there is some speculation that, especially when you're focusing on the parent compound cisplatin, today there are second-generation platinum-2 compounds that have been designed based on the parent compound cisplatin that have enhanced effect, but still, these drugs have very similar issues. And so DNA may not necessarily be the primary or the only cellular target for its cytotoxic behavior. One of the things that can hint at this is that it has been observed that the platination of the guanine residues is actually rather slow. And so efforts have been done in order to understand what are some other significant platinum biomolecule interactions. So they have a combination of targeted and untargeted approaches. What I mean by that are there are some omics approaches just to get a general sense of where platinum is being localized within the cellular environment to see if it's platinating proteins or where is that metal going. And then there are those targeted approaches where a particular biomolecule is isolated and the study is done to see whether that molecule can interact with platinum itself. So that's what a targeted versus an untargeted approach is, looking specifically at one biomolecule at a time or versus doing an assay to get a general sense of where a particular metal may be localizing. All right, so there is some suggestions that RNA could be a very important target of platinum. Okay, it is a nucleic acid, and so with this study that was done with these cells, was observed that platinum can bind more effectively to RNA than DNA, 4 to 20-fold times more. And so you can have similar types of adducts forming with RNA. There's also a suggestion that platinum can actually bind to a number of intracellular proteins or cellular proteins, which can have a detrimental effect. Okay, some of these can be detrimental not just to cancer cells, but also to just non-cancer cells. So here we have one possible target, the NHE1. So this is a membrane protein, sodium proton exchanger. Perhaps this being a membrane protein, it could be an initial target. It can cause lowering of intracellular pH, increase membrane fluidity, and apoptosis by the FAS pathway. We also have suggestions of platinum being able to affect the zinc finger proteins. And so if you recall last time when I discussed platinum being able to metallate albumin, I also mentioned that there is a capacity of platinum to affect the circulating zinc, because zinc can bind to albumin, and it's proposed to an extent that albumin can serve as a delivery vehicle for zinc. And so something similar can occur here, where platinum can affect the functionality of zinc finger proteins. So these proteins are the most abundant proteins in the eukaryotic genome. They have extraordinarily diverse functions. And so here I'm just giving you a list. DNA recognition, RNA packaging, transcription activation, regulation of apoptosis, protein folding and assembly, and lipid binding. Some of this can be even indirect in terms of the zinc finger protein providing a structural template to help facilitate these type of functions. But what has been observed is that the platinum has the capacity to displace zinc. So this is a type of transmetallation, and this type of transmetallation can be relatively fast. And so that substitution of one metal for the other, so the loss of the zinc now for binding of platinum, actually leads to observed inhibition of the function of these zinc finger proteins. So clearly, the platinum does not have the capacity to replace the function of the zinc. There's also the suggestion that tubulin is a potential target of the platinum. This is a 50 kilodalton protein that self-assembles to form microtubules involved in cell division when chromosomes separate during mitosis. And so it's been observed that platinum in this form, so a possible speciation of that metal, can alter the tubulin structure by binding to the N7 site of the base of the guanosine triphosphate. So this particular molecule is the energy source of this protein. So by metallating that triphosphate, that nucleotide, basically it is altering it to the point that perhaps it's not actually recognized by the protein. You also have the effect that the platinum can have on the thyrodoxine reductase system, so the TRXR. And so we're actually going to go back to this cellular target in a moment for another metal, gold. But this is a key enzyme in the disulfide dithiol regulatory system. It actually plays a very significant role in helping to regulate the formation of reactive oxygen species within the cellular environment. So it has been observed that the metallation of glutathione by the platinum from cisplatin can then lead to an inhibition of the thyroredoxin reductase system. And then a subsequent effect of this, and it's not entirely clear how this is manifested, but this effect can actually lead to hearing loss as a type of toxicity known as ototoxicity at high doses of the cisplatin. There's also suggestions that stress response proteins are affected by the presence of cisplatin, where there is platination of key proteins, which could impact regulation of the central metabolic pathway. And then there are some general organelle attacks that are a consequence of the presence of platinum in the cellular environment. So you have the mitochondria being affected by the presence of platinum, which can lead to an excessive formation. I probably should have wrote that, excessive formation of ROS, which could lead to oxidative stress. You also have degradation of lysosome. So lysosome is a compartment of the cellular environment that leads to protein proteolysis. So this is sort of a process of basically metabolizing proteins. But what's been observed is that the presence of platinum can lead to a release of excessive proteases. Okay, that's not a good thing. You also have stress that is impacted on the endoplasmic reticulum, which can lead to protein misfolding and even lead to deregulation of calcium. So you would have the effect of platinum on an essential metal, which is very important within our bodies. And platinum really plays a significant role in hormonal signaling. Right? So it's not entirely clear, right, that whether or not these are necessarily targets that enables the anti-cancer effect of platinum, or really if some of these are contribute to the toxicity of this metal. So it probably is a combination of both. Okay. All right, so with that in mind, I close my discussion about the understanding of the cisplatin compound and why there has been efforts to improve on this particular template, but even so, you still have platinum-2 compounds playing a significant role in terms of the chemotherapeutic regimens that are provided to people. But keep in mind that these are very toxic drugs. So they have their effect, but they're not necessarily something that is friendly to patients that do receive them. All right, so any questions? Feel free to post questions in the chat or to get on your mic. I'm going to switch to my other presentation, but if you do have questions, go ahead and just send them my way. Okay. All right, so as a transition then, we're going to look at two other metals today. We probably won't get through the second metal, but that's okay because we can continue this discussion on Thursday. We're still in the same theme of looking at anti-cancer approaches. So what I wanted to do was to present two different metals that have not necessarily advanced to the clinical market, but both of these metals have advanced to clinical trials for the application for an anti-cancer application. You will see that gold has actually advanced for the clinical market in terms of its application in the arthritis field. But what I wanted to do was to present a contrast here of two distinct metals in terms of their general inorganic properties, but also looking at their distinct intracellular targets as for you to get an understanding of how you might be able to appreciate metals in terms of facilitating the possibility of hitting different cellular targets. All right, so we're going to focus on gold compounds in terms of their potential anti-cancer properties. All right, so just a general perspective of gold. So gold predominantly can exist in the oxidation states of +1 and +3. And so that actually has a really a big impact in terms of the type of chemistry that and compounds that the gold can actually form. In the reducing environment of cells, gold will predominantly exist in the +1 state. And so now let's consider then the coordination chemistry of gold 1, as that can lead to an insight as to compounds that can be made with this metal. So when we're talking about gold 1, it has a completely filled an electronic shell with regards to the d orbital. So it is actually d10. And because it is d10, it has 0 crystal field stabilization energy. All right, so thinking back to our discussions about that, well, what does that really mean? Well, remember, we talked about crystal field stabilization energy as one of the factors that could be a thermodynamic drive for the formation of metal ligand compounds, right? The stabilization of the d orbital electrons can be a really a significant factor as to why metals will affiliate with ligand. Well, that factor does not play a role here, right? So the structure of gold 1 complexes is actually determined by steric effects of the ligand and simple electrostatic interactions between the ligands and the metal ion. And so here is just an overview of the general coordination geometries that gold 1 can adopt. So gold 1 is a soft Lewis acid. All right. So when you're now thinking about a perspective of metals and their anti-cancer function, so compared to platinum 2 is also a soft Lewis acid. So keep that in mind. All right. So gold 1 has 3 principal coordination geometries. It can have coordination number 2, 3, and 4. And so here I'm just giving you the idealized geometries that correspond to these coordination numbers. And so there is a preference for coordination number 2 when you're talking about anionic ligands. With regards to reactivity of these of gold 1, so here I'm just giving you a representation of how you can essentially ligate the metal center. So we're focusing here on the ligation of gold 1 with respect to these phosphine ligands. And so phosphine actually plays a significant role in terms of the story that I'm going to tell you about gold 1. And so here is just a suggestion of the type of ligand exchange that can occur, where you have the formation of 1 gold with 2 phosphines, and then the successive ligations of additional phosphines, leading to as many as 4 phosphines coordinated to that metal center. And so coordination number 4 for gold 1 tends to have a tetrahedral geometry. Interestingly, if you use P31 NMR, so you can actually track the phosphorus signal, right? So similar to what you would do for proton or carbon, or in this case, you're looking at the phosphorus group. What is very distinctive is that it demonstrates rapid exchange between ligands. Okay, so there are no distinct P31 peaks for this particular set of compounds, but rather a change in chemical shift as the gold and phosphorus ratio changes. So what I mean by that is that if you're trying to track the formation of the change of the gold phosphine from the coordination number 2 with 1 phosphine and all the way up to coordination 4 with all 4 phosphine group, it becomes quite difficult because in the NMR of this reaction, you don't get the distinctive phosphorus signals for each of these different species. Rather, you get sort of this broad signals where that as you change the ratio of the gold and the phosphorus, you do see a chemical shift, a shift that goes in the direction of perhaps favoring the higher order gold phosphine ligation. All right. But also very important is gold 3 in terms of what it can play in the understanding of general properties of gold, but also an anti-cancer property. So gold 3 is D8. It is isoelectronic with platinum 2, so that's actually very interesting. So it being a D8 metal, it has it typically will adopt a square planar geometry. But the crystal field stabilization energy for gold 3 is actually greater than the crystal field stabilization energy for platinum 2. So that can play a significant role. Gold 3 is an intermediate metal, unlike platinum 2, which we know to be a softer metal. But nonetheless, because of that intermediate property, it has the capacity to coordinate to a diverse set of ligands. So gold 3 can coordinate to thiol groups. It can also coordinate to nitrogen atoms, particularly we're focusing on the DNA bases. It uses associative mechanism of ligand substitution like platinum 2. Okay, so perhaps that doesn't make too much sense unless you took inorganic chemistry to really appreciate what I mean by that. But basically, the association mechanism is very similar to what I just showed you for the gold 1, the successive ligations potentially occurring through an associative mechanism. And redox agents can allow for both gold 1 and gold 3 to coexist under biological conditions. But I did mention to you that within the cellular environment, gold 3 would likely be reduced to gold 1, right? So just keep that factor in mind. All right, so historically, looking at gold in terms of its therapeutic application actually dates back to 3000 BC, where Egyptians used gold in purifying tonics. 2500 BC, the Chinese used gold in medicine and tinctures for healing. So it wasn't that they truly knew what the gold was doing, but there was an understanding that there was some sort of effect that the metal was having. In the 1st and 18th century, alchemists would actually use gold, and some of them would actually drink gold powders to enjoy longevity. In 1890, Robert Koch, he applied gold cyanide for tuberculosis. In 1925, 1935, gold was formally applied to treat tuberculosis, especially gold thiolates. And so here we have a representation of compounds, sulfonol and myocrystin, that shows the interaction of gold with thiol groups of these particular molecules. This is actually a sugar molecule, and that actually becomes really important in terms of our discussion in a moment. In the 1920s, excuse me, gold thiolates were applied for the treatment of arthritis. And so it is actually in the arthritis field where gold drugs really took off. And so we're actually going to return to this subject in a later lecture. But I want to introduce this history because it's very important in terms of establishing the importance of gold in an anti-cancer application. All right, so initially, anti-arthritic gold drugs were of the form, some of them, and when we're talking about gold, were of the form of gold 1 thiolates. And so the type of therapeutic they were intramuscular gold, they provided an intramuscular gold therapy. They fell under the category of a disease-modifying anti-rheumatic drug, so DMARD. And these, in terms of the administration, were weekly, monthly intramuscular injections, dosages of about 50 milligrams of gold. Okay, so here you have some compounds. So we've already seen 2 of these. This is another one that were examined for their anti-arthritic applications. So they do present toxicity. And here I'm just going to list one of them, but in our later discussions, we'll see that there's actually even more with regards to gold and toxicity. So one of the major ones being the nephrotoxicity, so toxicity of the kidneys. However, gold still, to an extent, is clinically used, but typically in combination with other drugs. So they're not necessarily frontline drugs or monotherapeutics, but they can be used in combination. So another class of anti-arthritic gold compounds are the gold phosphines. So here, gold 1 phosphines. One of the major ones was aranophen. And so this particular drug is orally administered. So here, about 6 milligrams per day. There's less excretion via kidneys, resulting in reduced gold retention. And it was actually introduced to the drug market in 1985. Okay, so this compound did advance to human use. The thing is that it is actually currently rarely used for reasons that I'll explain later on. But here I present to you the structure of Aranafin. So we have our gold 1 here in the coordination number 2, coordinated to a thiol group of this sugar molecule. So it's a thiol glucose is acetylated. So all the OH groups are acetylated. And then you have a phosphine group, this triethylphosphine, that serves as the other ligand of the gold center. So in terms of this drug and in terms of the field, there is a lot of interest in repurposing Aranafin because it actually displays a lot of very distinct properties that people want to exploit for other applications. So here's just a list of other diseases or conditions for which Aranafin has been examined. And you will see in a later lecture a discussion about, and even a recent examination of Aranafin for a potential anti-COVID-19 application. All right, but let's focus on the set of studies that have been done to try to repurpose Aranafin for an anti-cancer application. So this stemmed from the following, that classic anti-cancer drugs, 6-mercaptopurine and cyclophosphamide, have immunosuppressive and anti-inflammatory properties. These were exactly the same type of properties that were exhibited by gold compounds used to treat arthritis. It was also observed that arthritic patients receiving gold therapy had statistically lower incidences of cancer, which suggested that the gold drugs may actually have an anti-tumor effect. So it's a combination of that finding plus the known immunosuppressive and anti-inflammatory properties that really compelled people to examine the anti-cancer application of this compound. So it's been observed that Aranafin is anti-proliferative to many tumor cells or many cancer cells in vitro. This behavior, however, is reduced by the presence of serum proteins due to decreased cellular uptake of the gold. Studied in 15 tumor models in mice revealed that it really was active against the P388 leukemia type of cancer, and that was through an interperitoneal administration. A subsequent study, a structure activity study of 63 complexes of the general formula, so gold, with ligand L, ligand X, okay, revealed the following. It revealed that in an in vivo anti-tumor activity study, particularly against P388 leukemia, the optimized, or at least the compounds that have the optimal effect, again, an anti-tumor effect, were those in which there was a ligation to gold-1 with a substituted phosphine and a thiol sugar. And so Aranafin falls very nicely into that category because that is what you have, is a substituted phosphine and a thiol sugar coordinated to the metal center. All right, so this suggested that something about that type of coordination had the most potency against the set of tumors that were examined, the set of cancers that were examined. At submicromolar concentrations, Aranafin induces mitochondrial permeability transition. All right, so what does that actually mean? So basically what that means, it makes the mitochondria much more permeable, all right? And that subsequent effect of that is the release of the pro-apoptotic cytochrome C. So for those of you that are, for the review article, looking at drugs that can potentially synergize with cytochrome C or that use cytochrome C as part of its mechanism, well, Aranafin falls into that category because its effect of the mitochondrion leads to the release of cytochrome C, and that actually can lead to the induction of apoptosis. But another observation in terms of what Aranafin can do is that it can inhibit the thyrodoxine reductase. I already mentioned that enzyme briefly in our discussion of platinum. So it's been observed that the gold can actually have an inhibitory effect of this enzyme. So here I'm just showing you an IC50 concentration in terms of the inhibition of that enzyme of about 20 nanomolar. So it has been proposed that the gold one perhaps binds to the active site selenocysteine. All right, so let's give a visual as to this proposed mechanism, because that was words, but really, what are we actually seeing? So it's proposed that the aranophen, okay, is actually not a very stable compound, but in a later discussion, we'll go a little bit into the speciation of this compound. But here I'm just giving you the overview of what's speculated to be a mechanism of action, where the aranophen will deliver a portion of the molecule. So a part of it will be transported into the cellular environment, and is believed to be critical that the gold phosphine component crosses, and then that this compound can actually have two major targets within the cellular environment. There are actually two major targets that are very similar. So within the cytoplasm, it's believed that the gold impacts the thyroredoxin system, right, by inhibiting, for instance, inhibiting thyroredoxin reductase at its active site selenium. Okay, and so why would that be the case? Well, if you think about gold one, it is a soft acid, right? And selenium and the sulfur. Okay, so there's a really important groups that are present at the active site of this enzyme. Well, these are very soft bases, and so there is a natural preference for gold to be able to hit these sites, and in a consequence of metallating those groups, what it does, it affects the redox cycling that this particular enzyme facilitates. And it's also believed that the gold can affect thyroredoxin itself in a very similar capacity. So, what is the ramification of this effect? Well, the ramification of this is that you lose that control of this redox cycling process, and that can lead to then an excessive buildup of reactive oxygen species. And then that leads to a series of effects that can trigger cell death. In much the same way, it's been observed that the gold phosphine group can also affect the thyroredoxin system found in the mitochondrion. And so here we see a very similar effect as I proposed for what is observed in the cytoplasm, where now we're looking at the thyroredoxin 2 system being impacted in a much the same way, where the gold can bind to the selenium of the active site of the thyroredoxin reductase enzyme, or even bind to one of the thiol groups of the active site of the thyroredoxin system. And so, as a consequence of that, we get something that's very similar happening, where we have an excessive production of reactive oxygen species, and then that leads to what I mentioned previously, making the membrane more permeable. And as a consequence of that increased permeability is the release of pro-apoptotic factors like cytochrome C, which can then lead to caspase activation and then cell death. All right, so here you have that sort of the downstream effect of gold hitting these cellular targets. All right, but then what are the limitations of gold 1 complexes, especially these linear gold 1 complexes? What a major limitation is that they are very reactive. And so because of that reactivity, what can happen is that you have ligand substitutions that occur extremely quickly that essentially will decrease the activity of the gold center. All right, so here's just a representation of an associative mechanism of an entering group that will then lead to a dissociation of a ligand and transform that compound. But perhaps that compound that is generated may actually affect the lability of the gold to be able to hit a certain target. For this reason, there have been efforts to try to go around this limitation by using other types of structures of gold 1 compounds. And so, for instance, the use of bridging ligands to help to decrease this ligand exchange, because the thing is that it's not that you necessarily want to stop ligand exchange from happening, because you do want that metal center from hitting a target. What you want to do is to try to decrease how labile the ligand exchange is to then boost the ability of the gold to hit a particular target. And so you don't have then, with these type of complexes, you don't have the capacity to go through an associative mechanism. Instead, what has to happen is a dissociative mechanism has to take place, where perhaps one of these chelators, part of the chelation gets lost, and so you have partial coordination of a ligand that then leaves a coordination site available to which an entering group, or a nucleophile, can bind to a metal center. So you're still enabling ligand exchange. What you're doing is you're trying to decrease this rapid exchange that would potentially decrease the activity of the gold itself. All right, so let me clear that up so you guys can see the take-home messages there, where you have the linear gold compounds will have a high thioreactivity, thus decreased antiproliferative property, whereas the use of those other type of ligands that are chelators, you have a low thioreactivity and optimal lability to decrease loss of activity. All right, so let's explore some of these chelators that have been used. And so here we're looking at the use of bidentate phosphine ligands. And so here's a representation of one, this DPPE, and you get the formation of these gold tetrahedral complexes with those bidentate phosphine ligands. Okay, so this particular compound that you see to the right has demonstrated a significantly broader range of activity than eranofin and its analogs against a number of different tumor models implanted in mice. It is quite stable in the presence of thiols and blood plasma. All right, so that's actually important because you don't want to have that rapid binding of thiol groups or even proteins to the metal center that may affect its ability to be potentially delivered into cells. But nonetheless, there's still toxicities associated with these type of compounds. What has been observed are acute cardiac, hepatic, and vascular toxicities. It's still believed that the primary mechanism of action of the gold of these compounds is via mitochondrial dysfunction. So again, affecting the thyroreducin system, which would then affect the mitochondria. Right, but let's look at some other examples of these compounds. So here we still have that DPPE ligand, and then we have another related type of bidentate phosphine group. And so what we're seeing is the use of these two primary compounds with different substituents. And so these are the substituents. So there are different aromatic groups. All right, so let's look at the differences between compound to the left, this one, versus the compound to the right. Okay, and so we're looking at gold DPYPP versus gold DPPE. All right, so I'm going to ask you guys to look at these slides and tell me, what do you see? So the top slide is the effect of the compounds on the cancer cell line. Is a human breast adenocarcinoma, MB468, and the bottom is an effect on a non-cancer cell line. So this is a human mammary epithelial cells, HMEC. All right, so take a moment, look at this data, and either on the chat or you can get on your mic, what differences do you guys observe? All right, so I believe Daniela and Zayomi have hit it in terms of the observations here. And so Andres is inversely proportional effect in cell growth. So what we're seeing here is where both of these compounds, they're, generally speaking, they're very similar. They are tetrahedral compounds with bidentate phosphine groups. Really, the differences, it's not so clear from the actual figures which substituents, but presumably the same substituents for both. But we're looking at the core. So something about the backbone of those compounds is a difference, really. And so both of them are quite effective against in terms of the anti-proliferative effect that they have, where you have this dose response. As you increase the concentration, the cell growth is decreased, and it seems to be in a very comparable manner. But then when you focus on the non-cancer cell line, you see that the gold DPPE, so the compound to the right, that actually has also an anti-proliferative effect. And that's not necessarily a good thing, right? But the other compound does not actually have that anti-proliferative effect. We generally have that the cell growth is unaffected. Maybe at the higher concentrations, there's a little bit of an increase of the amount of those cells. Okay, so for whatever reason, something about the compound with the ligand D2PYPP results in that effect. Okay. All right, so looking at then, that's sort of the overall, like what impact do these compounds have on cell lines? But now looking deeper, remember I talked to you about this idea of focusing on a targeted effect. All right, so the thyroreductin reductase inhibition study was performed to get a general sense about whether or not these compounds, or a particular compound in this case, so the D2PYPP, that one, which had, remember, no effect on the non-cancer cell line, but an antiproliferative effect on the cancer cell line. So then they looked at this further, again, focusing on both cell lines. And so they wanted to see how the compounds were impacting on the thyroreductin and thyroreductin reductase proteins, so both of them. And so we have control, subject control, as I mentioned to you in our last lecture, typically is just the media, the buff that you're using, versus the compound. And then we're looking at both cell lines. The black one is the cancer one, the white one is the non-cancer one. And so what we can observe here is that the compound is actually affecting the amount of the thyroreductin system, the thyroreductin protein, much more. So you can see here the activity is decreased relative to control, much more in the cancer cell line than in the non-cancer cell line. Okay, and then as far as having an effect on the thyroreductin reductase protein, well, the same thing is true, probably to a lesser extent, in terms of affecting that enzyme more in the cancer cell line versus the normal cell line. All right, so it looks like that not only are we seeing the global effect of hitting the cancer cells much more than the non-cancer cells, but also that you're actually hitting the proposed targets. So let me clean up this so you guys can see my notes here. So this was an experiment where cells were incubated with 5 micromolar of the compound, 2 hours, 37 degrees. The cells were lysed, and then the activity of thyroreductin and thyroreductin reductase activity was examined. The thyroreductin reductase has a selenocysteine, so that is cysteine substituted, or the sulfur substituted with the selenium in one of its 2 catalytic sites. So the presence of the seleno group actually makes that base even more soft than the thiolate group. Okay, and so it's believed that inhibition can result in apoptosis. All right, so that's a really nice finding there. All right, so here again, I'm just showing you another representation of the gold compound, but from this perspective, this particular compound entering the cellular environment and appearing to be able to hit the same target. So what I showed you previously was sort of a general mechanism for the gold phosphine from aranophen, but here we're looking at it from the perspective of that gold compound with the bidentate phosphine groups. And so again, we're seeing the dissociation of one of the arms of the chelators, allowing for the nucleophiles, so the active sites, uh, selenocysteines, or even the thiol groups, to bind to the gold, and in that vein, being able to inhibit the mechanism of these proteins, whether it's in the mitochondrion or whether it's in the cytoplasm. All right, so here again, the same picture, but now we're just saying this being true generally for gold compounds. And for gold 3 compounds, possibly, if you think about the gold possibly being reduced to gold 1 and then being able to have this effect. That gives us kind of the overall picture of what's possibly happening. All right, but let's explore other types of ligands. And so people have explored the use of carbenes. So N-heterocyclic carbenes, NHCs, can serve as alternative ligands to phosphines. So here we have the representation of an imidazolium cation that in the presence of base will become this imidazole 2-idylidene. Okay, and so we have our carbene group right here. Okay, so NHCs are electron-rich, neutral sigma donor ligands. Electronics of these type of ligands can be altered by changing the nature of the azyl ring substituent. So those R groups can be altered, and that can have an impact in terms of the electronics, really the effect of those electrons interacting with the metal center. So the NHEs can form very strong bonds with metals, even stronger than phosphines. So here we're looking at the formation of gold compounds with these carbenes. So you have the formation of these type of complexes. Now you're going back to a linear compound. All right, so we talked about the limitations of those compounds. Remember, so linear gold 1 compounds tend to undergo associative mechanisms which can rapidly decrease the activity. Well, in this particular study here, what was done was through the use of the R groups were to create these sort of wing tips that have the capacity to basically protect the environment around the gold. All right, so basically serving as a way to decrease the accessibility of the gold site, and in that respect, decrease that rapid ligand exchange. Right, but again, you don't want to eliminate the ability to ligand exchange. That's not the goal here. It's simply to slow it down. And so here we have then this illustration of the gold being able to interact with the thiol groups, the seleno groups, and being able to then generate the formation of these type of compounds. The cysteine groups interacting with the gold itself. Right, so just sort of a proof of principle that this type of complexation can happen. All right, so then studies with these compounds, and so here we're looking at the effect of the family of compounds against this breast cancer cell line, someone that we looked at before, the MDA-MB1. And so we're looking at all 3 of them having an anti-proliferative effect. We see this from low micromolar, or actually you're dipping into nanomolar here, all the way up to 100 micromolar. So I would say probably not all 3 are that great, but generally speaking, you have this anti-proliferative effect. But then over here, we're looking at the impact of the gold compound on 2 proteins, the thyroid reducin reductase, and then this other protein, this glutathione reductase. All right, so what do we see here? Well, we see that there is an inhibition, a concentration-dependent inhibition of the thyroreducin reductase, but the glutathione reductase is not being inhibited by this compound. All right, but why am I even bringing up, or why did they even study this other enzyme? The reason why they studied this other enzyme is that there are similarities between the glutathione reductase and the thyroreducin reductase. So the GR, glutathione reductase, is a homodimeric enzyme that is responsible for reducing the oxidized form of glutathione to its reduced form, right? So what you see a lot in the cellular environment is glutathione transforming into its oxidized disulfide bridge form into its reduced form. So it's back and forth, and that's how it can help facilitate the redox processes within the cellular environment. All right. The selectivity for thyroreductin reductase versus the glutathione reductase is not entirely apparent, but the glutathione reductase does not contain a selenocysteine at its active site, but does contain a cysteine. So perhaps that difference may be a difference enough for the gold of this particular compound, the carbene compound, to be able to react with one side versus the other. Again, that's not entirely clear, but that's just the illustrations here to give you a perspective of these two enzymes, and that what was observed was that the thyroreductin reductase system was what was actually impacted on by the presence of that gold center. Okay. All right, so that gives us an overview of the gold 1 compounds. We looked at the aranophen, we looked at the second generation compounds with bidentate phosphine groups and even carbenes. So now, just very briefly, we're going to talk about gold 3. So I gave you a bit about a background about gold 3 to distinguish it from gold 1. So gold 3 is isoelectronic and isostructural with platinum 2. However, it is not stable toward reduction like platinum 2 is, so gold 3 can be reduced to gold 1. As an intermediate acid, gold 3 is believed to bind to the intermediate nitrogen donor groups of the DNA bases, so it does have that capability. And it's been observed that it can bind to the guanine bases, most likely in a monoatomic form, okay, as opposed to an inter- or intrastrand crosslink. So in other words, gold 3 can potentially act like platinum 2 in terms of being able to metallate the guanine bases of DNA. Right, so there is that capability. And so what people have tried to do is, to an extent, is to exploit that similarity with cisplatin. And to be able to do so, remember, if gold 3 is very reduction prone, right, what has to happen is you have to design a compound to be able to minimize that capability of the gold 3 to get reduced to gold 1. So these compounds have been designed to stabilize the trivalent oxidation state and incorporate labile sites for the necessary interaction with biological targets. All right, that's not an easy thing to do. So chelators are used with sigma donation capability. And then you have ligands in the cis position that are easy to displace, analogous to cisplatin. All right, so here is just a representation of the formation of these type of compounds. For instance, we have this compound here, where you have a chelator, right, that's coordinated to the gold, and you have then these two chloro groups, right, so that you form something that's analogous to cisplatin, because then potentially those chloro groups can't dissociate. And so you have something very similar over here via the use of these chelators that have a sigma donation capability. And then over in this example, others have exploited the macrocyclic effect, where the use of these type of groups that will help to stabilize the gold in the center. All right, so it provides a ligation that is enable the coordination number 4. So this particular compound, this gold TTP chloride compound, has high anti-cancer activity with IC50 values in the sub-micromolar range against cisplatin and multi-drug resistant cells. Like other gold 3 complexes, the metal ion may be able to target DNA, so then it may be the case, but nonetheless, it's been observed that the main mode of action appears to be mitochondrial targeting of the thyroreductin system. And how is that is not entirely clear. Perhaps the gold center does get reduced to gold 1, and somehow then that can facilitate the interaction of the gold to the thyroreductin system. Why? Well, if you think about it, if this is in fact how the gold is coordinated, well then in the axial positions, the if you think about this as something that is forming sort of a square planar arrangement, there is then axial positions available for potential ligation, perhaps by the active site groups of the thyroidexin system. All right, so that's not entirely clear, but it's just a speculation that I propose. All right, so that gives us then a perspective about gold. And so gold, in terms of its oxidation state of 1 and 3, we're talking about metals that fluctuate between a soft acid and a intermediate acid, but then the primary cellular target is believed to be the thyroidexin system, whereas platinum from cisplatin is believed to target primarily DNA, although I did show you a series of complexities to that idea of what platinum actually targets. All right. Again, if you have any questions, feel free to post your questions in the chat. Well, now I'm going to transition to then discussions of another metal that also presents a different type of chemistry and also different type, a potentially different type of target. All right, so we're going to focus on titanium-4 compounds in terms of their potential anti-cancer effect. And so the reason for this is that titanium-4 is, in large respects, a big difference, a different type of metal ion versus platinum and even gold-1 and gold-3. So when we're looking at titanium in our environment, it actually can exist in a variety of oxidation states. All right, so here's just a perspective of -1 up to a +4 oxidation state. But predominantly, what you're going to see is that titanium-4 is the most stable and common oxidation state. And that is because many of these other oxidation states can be readily transformed to titanium-4 in the presence of air and water. One of the most predominant forms of titanium is titanium dioxide. It is ubiquitous, okay, and it can exist in either amorphous form or in a very stable crystallized form. Here I give a representation of crystallized forms in the anatase, the brookite, and the rutile crystal lattices. All right, so that is where you can find titanium abundant in terms of its crystallization. Even within the reducing cellular environment, titanium is expected to exist in the +4 state. And that is because within the cellular environment, there simply isn't a strong enough biological reducing agent to reduce that titanium-4 to titanium-3, right, or even further down. Another really interesting thing is that titanium-4 is D0, meaning that it does not have any d orbital electrons. So it is diamagnetic, and like gold-1, it does not have any crystal field stabilization energy. Right, so gold-1, D10, titanium-4, D0, both no crystal field stabilization energy, right? Well, because you're not going to stabilize any d orbitals if there are not even electrons in there anyways. Lack of crystal field stabilization energy should make these compounds ligand exchange labile. However, the high oxidation state decreases this lability, right? And so that's actually important. So here I present to you a table that shows the types of coordination number that titanium can adopt in terms of complexes and the corresponding geometries. So coordination number 4, tetrahedral. So I showed you guys this compound in an earlier lecture, this titanosyne dichloride, actually going to be an important compound in our discussion today. And so you see the other types of coordination numbers that titanium can adopt. One of the primary ones is actually coordination number 6. And so you get geometry octahedral, which I would say realistically distorted octahedral. So here I present to you a bit of the bioinorganic chemistry of titanium. So titanium is a hard Lewis acid. It has the capability of a preference for binding to nitrogen and oxygen groups. And here are some of the sites to which titanium can coordinate in terms of biomolecules. So just to give you the overview of the types of compounds that you potentially can find titanium forming with biomolecules. I'm not going to go through this in any significant detail. Here are some biomolecules which have those very same moieties that I showed you in the previous slide that have been shown to have the capability of binding to titanium. Now, capability does not mean that you actually see this happening in the in vivo context, right? That's two different things. But here are just an overview of compounds that people have actually studied in terms of their ability to bind to titanium. All right, but something that I really want to communicate is that there is a similarity, is not exact, right? A similarity between the biocoordination of titanium and of iron. And here's just a representation of the similarity, but also some interesting differences between how titanium can bind to catechol. And so we talked about catechol in terms of that is a metal binding moiety that you find in siderophores. And so we're looking at the catechol ligand as it speciates with titanium, and then how it speciates with iron. So they're actually very similar in terms of the type of complexation that we see. We see very similar 1-1 metal ligand interactions, 1-2 metal ligand interaction, and 1-3 metal ligand interactions. But if you look closely, there are some actually really important differences. So we can look at this from the perspective of the pH-dependent speciation. And so here we have the speciation that corresponds to iron 3, and then below we have the speciation that corresponds to titanium 4. So above, what we see is that at a very low pH, you have iron basically in its aquated form. But then once you start going above a pH of 2, okay, then that catechol ligand has the capability of chelating that metal, right? And so then you go through the series of complexes 1-1, 1-2, and 1-3 that can form, where that 1-3 compound really plays a factor above a pH of 8. So titanium shows something very similar, but there's actually a lot of important differences as well. At a pH as low as a pH of 0, you see that titanium is already, it exists as a hydrolyzed species. So what I mean by that is that water molecules that are bound to the metal are already deprotonated. So that is a very, in terms of its Bronsted-Lowry acidity, it is a strong acid. And then you start to see the formation of compounds with the catechol in the acidic range. But already by a pH of 4, you start to see that the catechol ligand can actually saturate the coordination sites of titanium. So that's at a pH of 4 is where you start to see the presence of that compound. Whereas for the iron case, that you observe above a pH of 8. So that shows us some really profound differences in terms of the ability of these metals to coordinate to these same type of ligands and form the same type of compounds, but at a different pH value. And so that speaks to the difference in the actual Lewis acidic properties of these two metals. Okay. So I said that the coordination is similar. And so as you saw in the previous slide, similar is correct, but here is where titanium and iron 3 can actually deviate very significantly. So here we see that the redox chemistry is different between titanium 4 and iron 3. And so what we're looking at here is a correlation plot that was established for different ligands. So these are different types of ligands that have the capability of forming titanium compounds and also iron compounds. And so, as an example, here we have a ligand, H-bed, and so I'm actually not going to show you the structure, but that is a hexadentate ligand. So it has the capability of binding to these metals at 6 different sites. So if you look at the correlation, if you were to see the reduction potential of the titanium compound of that ligand versus the reduction potential of the iron compound, what you would notice is that systematically for all these ligands, the iron version has a lower reduction potential versus NHE than the titanium does. Okay, so a lower reduction potential. So what does that mean? Well, typically what that means is that many of the titanium versions of these compounds will fall at too negative a reduction potential so that the titanium 4 will remain as titanium 4 versus the iron 3, which has a higher reduction potential. Generally speaking, many of these iron compounds will have a reduction potential that will facilitate the ability of the iron 3 to be reduced to iron 2. And so that actually highlights a major difference between both metal ions, that especially at a pH of 7.4. So this is going to be very true at a pH of 7.4. That iron 3 tends to be redox active, but titanium 4 is redox inert. Redox active. Okay, and that's actually really important because that can really distinguish between a lot of the properties that one metal can facilitate versus the others cannot. The other cannot. All right, another really important feature about titanium. So to an extent, we've already seen this, that titanium is hydrolysis prone. So here's just the representation of this hydrolysis process for titanium, leading ultimately, if you think about this downstream, especially at a really high pH, to the formation of titanium dioxide. With regards to how this impacts the solubility of titanium, if we're focusing on a pH of 7.4 and focusing on a species that has some amount of solubility, for instance, this titanyl dihydroxide species, well, if you were to measure its solubility product and then to calculate the solubility of the titanium, we're talking about a solubility that's really only femtomolar amounts. So really, titanium, if you were to dissolve it at a pH 7.4 water, that titanium would be in so low concentration that essentially would be biounavailable. As it turns out, the interaction of titanium with in the biological environment with metal chelators, such as small molecules, such as you're talking about citric acid or tunochrome, or even proteins, for instance, transferrin, what happens is that you can significantly alter the solubility of titanium such that the titanium is present at significantly higher concentrations in human blood than one would expect. So it has been observed that titanium can reach as high as nanomolar concentration in human blood. So we look at the difference between what we would call the innate solubility versus the solubility as influenced by the presence of biomolecules. You can see that there's many orders of magnitude different, and that orders of magnitude different can facilitate that titanium is bioavailable. Okay? Again, bioavailable does not mean that it's actually functional, but it could have the capability of being functional. All right, so in the human body, it's been proposed that the protein, the iron transport protein, human serum transferrin, actually plays a significant role in the uptake of titanium released into the bloodstream. And this is actually particularly true for people who have prosthetic implants that are titanium containing, that the metal, some of the metal that is leached from these implants can be captured by serum transferrin, and then the protein can become metallated and can help to the transport of this metal. So recently, I would say within the last 4 or 5 years, insight into the coordination of titanium by this protein has been established. And so here is the first crystal structure that was obtained for titanium bound to any protein, but in this case being the serum transferrin protein. What was observed was that metal can coordinate to this metal binding site in a manner that's very similar to how iron coordinates, right? But there was actually a really important observation that was seen in this particular crystal structure, where there is an additional ligand present at the metal binding site of that titanium, and that ligand is a citrate molecule, which suggests that citrate may actually play some sort of metal transporting role in the human body, or even in serum. And so that's actually significant because prior to this study, citrate in terms of its metal transport in the human body was really not something that was well established or even studied. But citrate does really have the capability of serving as a transporter, especially for bacteria. But here, this study shows the potential for citrate to interact with serum transferrin in at least regulating the solubilization and even the transport of titanium. And so, just to highlight the similarities, but also differences between how iron is coordinated to this protein and also how titanium is coordinated. And so, what we observed is similar ligation to the tyrosines of the metal binding site of this protein, also the presence of carbonate, but a big difference being that these two amino acids that are present for iron, at least in that crystal structure, was observed that it was substituted by the presence of the anion citrate to facilitate coordination of the titanium. All right, and so that has led to speculations of, well, what is the route of titanium transport by the serum transferrin protein? And so here we see a postulation of titanium citrate coordinating to the transferrin protein, okay, and then that getting recognized by the receptor, and then possible endocytosis can bring the titanium into the cellular environment. And so a combination of the decreased pH of the endosome plus the presence of some sort of intracellular ligand can lead to ligation of titanium and then release of the titanium into the intracellular environment. So I'm going to stop right here, because then this actually leads to a nice discussion as to then what can affect the bioactivity of titanium and potentially what can regulate the anti-cancer effect of titanium. All right, so we will continue on from this point in our next lecture. All right, everyone, so I'll see you on Thursday. Take care.
Thank you, Professor. Can I ask you a couple of questions after the rest of the people leave?
Sure, that's fine.